According to the history books, cosmonaut Yuri Gagarin was the first human to ever go into space in 1961. A year earlier, however, Henrietta Lacks had gone up into space in the form of HeLa cells. At the dawn of the space race between the US and Russia, scientists did not know how being in space would affect humans on a cellular level. To find the solution to this problem, cells were sent on the early unmanned and manned missions into space. The first ever mission that sent a man into space was no exception. Microbiologist N. Zhukov-Veresnikov figureheaded the team that oversaw the sending of HeLa that he acquired from America into outer space. He sent them up first on the unmanned Korabl Sputnik 2, and then with Gagarin in the Vostok 1's craft.
On the morning of April 12, 1961, Yuri Gagarin and the cells were launched into space. Out of 200 Russian Air Force pilots, Gagarin was selected for the mission. Regardless of his piloting skills, the craft was designed to be mainly controlled either by a pilot on the ground or automatic controls. As it was not certain as to how the space would exactly affect the human body, it did have onboard piloting controls. Though these were very limited, with all this being so, protocol was broken and Gagarin was given control of the craft before launch. These onboard controls were only to be used in case of emergency. Going around the Earth once in 108 minutes, the craft's orbit peaked at 327 kilometers, or 203 miles, above the surface of the Earth.
These test runs proved to be useful, as they served their purpose and proved scientists with information on the nature of cells in space. While normal human cells were found to divide normally in space, the cancerous HeLa cells that were sent up showed scientists that cancerous cells would behave differently when exposed to the environment of space. When they went into space, HeLa cells began to divide more rapidly, with later tests showing that the rate of division would increase with each trip to space.
What is bioengineering? Bioengineering applies technology to the sciences of biology, which allows humans to control aspects of the body that had previously been controlled by nature. This branch of science can range from things such as artificial limbs, like the one in the picture on this slide, to going so far as to the altering of a person's genetics.
Bioengineering was a field that was relatively small pre-World War II, with most bioengineers falling under the category of agricultural and some chemical engineers. This would all change, however, after the war, thanks to the strong desire for medical advancements to be made. Because of this desire, bioengineering took off, specifically bioengineering in the medical field.
Along with many advancements that were made in bioengineering came many disagreements over the morals of altering the natural human body. For example, the use and alteration of tissues has brought moral and legal questions into the light. Damaged tissues and organs are restored using other substitutes that can be placed in a body. This led to many debates rising over legal ownership over tissues and whether or not people actually own their samples once they are removed from the body, as well debates over the potential profits that can be made from human tissue.
In bioengineering, no branch leads to more ethical debate than genetic engineering. Disagreements on whether or not it is right to change the natural way a human is made from their genetic information divides the scientific community. This issue can become especially heated when religion becomes involved. If we choose to set aside our differences in beliefs, however, then mankind can come to a just solution that we can all agree on.